Lab-grown organs go rogue, develop brain cells. Sean Adeltabatabai, your News Punch reports, lab-grown organs have begun suddenly sprouting brain cells, according to scientists at the Washington University School of Medicine. How is that possible? The miniature kidneys, which were made using test tubes in a lab, did not behave in the way researchers anticipated they would behave. Daily Star reports, instead of growing into different kinds of kidney cells, they in fact formed brain and muscle cells. The miniature kidneys, also known as kidney organoids, initially come from human stem cells and could eventually be used to treat people with kidney diseases. But the findings have gone rogue. Coldest back the research, with experts saying the fact that they behave in this way indicates the technique used to grow them from stem cells are actually creating other cells. Benjamin D. Humphreys, the director of Division of Nephrology, nephro means kidney, it's a Greek word, Division of Nephrology at the Washington University School of Medicine in St. Louis, said, "There's a lot of enthusiasm for growing organoids as models for diseases that affect people. But scientists have not fully appreciated that some of the cells that make up these organoids may not mimic what we would find in people. The good news is that with a simple intervention, we could block most of the rogue cells from growing. This should really accelerate our progress in making organoids better models for human kidney disease and drug discovery, and the same technique could be applied to targeting rogue cells in other organoids. Developing kidney organoids is driven by the reality that we have so many patients with failed kidneys and no effective drugs to offer them." Dr. Humphreys said between 10 to 20% of cells in the mini kidneys missed cues to develop further into kidney cells and instead becoming brains and muscle cells. When they pinpointed the stage this happened, they were able to reduce that quantity in research that could help other scientists eliminate these types of rogue cells in their experiments. Quote, "Progress to develop better treatments for kidney disease is slow because we lack good models," he said. Humphreys went on to add, "We rely on mice and rats, and they are not little humans. There are many examples of drugs that have gone and done magically well at slowing or curing kidney disease in rodents, but they failed in clinical trials. So the notion of channeling human stem cells to organize into kidney-like structure is tremendously exciting because many of us feel that this potentially eliminates that lost in translation aspect of going from a mouse to a human." This by Sean Adeltabatabai, Editor-in-Chief at Your Newswire, and this is on News Punch. Kindly support my Patreon account, since that's the only way I can continue what you like seeing on this channel. Thank you. You'll find it in the description box below.